ሃይ ሰላም ውድ ተማሪዎች እንዴት ናችሁ? እንዴት ቆያችሁ? ዛሬ በዚህ ቪዲዮ የምንመለከተው የሪሜዲያል ባዮሎጂ ከዩኒት 1 የተውጣጡ ጥያቄዎችን እንመለከታለን። ከ32 ወደ 32 የሚጠጉ መልቲፕል ቾይስ ጥያቄዎች ናቸው። ቤዝድ ኦን ዩኒት 1 the science of biology ከሚለው ምዕራፍ የተውጣጡ ጥያቄዎች ናቸው። ከዚህ በፊት the science of biology የሚለው ምዕራፍ በቪዲዮ ቻፕተሩን ተመልክተናል። ከዛ የተውጣጡ ጥያቄዎች ናቸው። በዋናነት እነዚህ ጥያቄዎች ለሪሜዲያል ናሽናል ኤግዛማችሁ እንዲያግዛችሁ ነው ያዘጋጀንላችሁ። ጥያቄዎችን አብረን እንሰራለን። እናንተ ቪዲዮ ፖዝ እያደረጋችሁ ለ5 ሰከንድ በኋላ የኔ መልስ ትከታተላላችሁ ማለት ነው። ጥያቄ አስተያየት ካላችሁ ኮሜንት ላይ ጻፉ። ፒዲኤፍ የምትፈልጉ ከሆነ ቴሌግራም ላይ እዚህኛው ሊንክ ላይ ባለው ቴሌግራም ሬሜዲያል ፎከስ 2018 በሚለው ሊንክ ላይ ገብታችሁ እዛ ላይ አክሰስ ማድረግ ትችላላችሁ። ቪዲዮን ላይክ ለጓደኞቻችሁም ሼር ማድረግ እንዳትረሱ። ኦኬ let's begin. First question, which of the following sequences correctly represented the transformation of a hypothesis into a scientific law? A, hypothesis, law, theory, principle. B, hypothesis, theory, law, principle, law or principle. C, law, theory, hypothesis, fact. D, theory, hypothesis, law. የትኛው ነው ትክክለኛ መልስ የሚሆነው? answer must be B, hypothesis, theory, law, principle የሚለው ትክክለኛ መልስ ይሆናል ማለት ነው። Number 2, what is the purpose of control group in an experiment? A, to test the independent variable. B, to be the untreated group for comparison. C, to recurse the data. D, to formulate hypothesis. The correct answer is to be untreated group for comparison የሚለው ትክክለኛ መልስ ይሆናል። Number 3, in the scientific method, what is the role of gathering information? To form conclusion, to design an experiment, to formulate a hypothesis. D, to analyze data. In scientific method, what is the role of gathering information? The correct answer is to formulate a hypothesis. ግልጽ ያልሆነላችሁ ጥያቄ አስተያየት ካላችሁ ወይም ኮሬክሽን ነው ብላችሁ የምታስቡት ነገር ካለ ኮሜንት ላይ መጻፍ ትችላላችሁ። Number 4, which of the following is an example of a dependent variable? A, temperature is an experiment. B, the rate of growth of seedling at different temperatures. C, the amount of light given to a plant. D, the type of soil used for growing plants. ከነዚህ ውስጥ example of dependent variable የሚሆነው the rate of growth of seedlings at different temperatures የሚለው ትክክለኛ መልስ ይሆናል። Number 5, which of the following is a field tool used by biologists? Mortar and pestle. B, microscope and microscope. C, plant press. D, centrifuge. Answer should be plant press. A biologist need to examine the surface detail of an insect in 3 dimensions. Which type of microscope would be the most suitable for this purpose? A. Transmission electron microscope. B. Compound light microscope. C. Scanning electron microscope. D. Phase contrast microscope. In 3D dimension, answer must be scanning microscope, scanning electron microscope. Number 7. What is the function of the objective lens in a microscope? A. To adjust light intensity. B. To hold the slide. C. To magnify the image. D. To focus the specimen. Function of objective lens in microscope: to magnify the image. Okay. Number 8, sorry. What is the difference between inductive and deductive reasoning? A. Inductive reasoning goes from general to specific, while deductive reasoning goes from specific to general. B. Inductive reasoning goes from specific to general, specific to general, while deductive reasoning goes from general to specific. C. Inductive reasoning is used for forming hypotheses, while deductive reasoning is used for testing hypotheses. D. Inductive reasoning is based on observation, while deductive reasoning is based on experimentation. The question: what is the difference between inductive and deductive reasoning? B. Inductive reasoning goes from specific to general, while deductive reasoning goes from general to specific. Number 9. Which of the following is a special branch of science that deals with living things? A. Physics. B. Chemistry. C. Biology. D. Geology. Which of the following is a special branch of science that deals with living things? Answer should be C. Biology. What is one of the main uses of biology? To study non-living things, to prevent and cure diseases, to build bridges, to study the stars. Main uses of biology. One of the main uses of biology: to prevent and cure disease. 11. Which type of microscope is used to study the surface of objects in 3-dimensional details? A. Transmission electron microscope. B. Compound light microscope. C. Scanning electron microscope. D. Simple microscope. Which type of microscope is used to surface of objects in 3-dimensional detail? Obviously, scanning electron microscope. Consider the scientists using a compound light microscope to observe a cell. They increase the magnification to the highest setting, but the image becomes blurry and they cannot see any more detail. This is due to A. The limitation of electron microscope. B. The resolving power of the microscope. C. The magnification of power of the objective lens. D. The interference of light waves. is due to the resolving power of microscope. B. Which of the following statement best describes the nature of scientific knowledge? A. Absolute and unchanging. B. Relative and continually tested. C. Limited to the laboratory. D. Based purely on opinion. Scientific knowledge, nature of scientific knowledge. Relative and continually tested. B. 14. In the scientific method, what is the role of gathering information? A. To form a conclusion. B. To design an experiment. C. To formulate hypotheses. D. To analyze data. Scientific method, what is the role of gathering information? To formulate hypotheses. What is the importance of having control variables in an experiment? Having controlled variables. A. To change multiple factors at once. B. To ensure the experiment is unethical. C. To keep conditions constant that could affect the outcome. D. To make the experiment more complicated. Having control variables. Importance of having control variables in an experiment. To keep conditions constant that could affect the outcomes. Answer C. 16. Which of the following is not a laboratory tool? Not a laboratory tool. A. Mortar and pestle. B. Insect net. C. Pipette. D. Petri dish. Not laboratory tool. Insect net. 17. Question number 17. What is used to magnify objects in a compound microscope? A. Mirrors. B. Single convex lens. C. Two convex lenses. D. Electron beams. Used to magnify objects in compound microscope. C. Two convex lenses. Which of the following best describes the interrelation of biology with scientific method? A. Biology is a science that relies solely on theoretical knowledge, disregarding the scientific method. B. The scientific method is a tool used in biology to study the nature and the functions of living things and their interactions. C. Biology is a branch of science that uses the scientific method only to study non-living things. D. The scientific method is not applicable to biology as is purely observational. The correct answer is biology with the scientific method. So the scientific method is a tool used in biology to study the nature and the function of living things and their interactions. 19. What is magnification in microscopy? A. The ability to distinguish between two points. B. The process of staining cells. C. The increase in the size of the image. D. The use of electron beams. Magnification. What is magnification in microscopy? The increase in the size of the image. C. 20. How does an electron microscope differ from a light microscope? A. It uses light instead of electrons. B. It has lower resolving power. C. It uses a beam of electron instead of light and has a greater resolving power. D. It can only view living specimen. How does an electron microscope differ from a light microscope? C. It uses a beam of electron instead of light and has greater resolving power. 21. Which type of microscope is used to study the surface of an object in three-dimensional detail? A. Transmission electron microscope. B. Compound light microscope. C. Scanning electron microscope. D. Simple microscope. Which type of microscope used to study the surface of objects in three-dimensional detail? Answer should be scanning electron microscope. What is a limitation of electron microscope? A. They cannot magnify objects. B. Living cells must be killed before they can be observed. C. They have low resolving power. D. They are used to study large objects. Limitation of electron microscope. Limitation of electron microscope የዘረዘር ነው የትኛው ነው? Living cells must be killed before they can be observed. This is the limitation of electron microscope. How does a theory become a law or principle? A. It is voted on by scientists. B. It is never possible for a theory to become a law. C. When a theory has proved invariable under all circumstances. D. After 10 years of acceptance. How does a theory become a law or principle? When a theory has proved invariable under all circumstances. C. What is the purpose of evaluation step in scientific method? A. To skip repeating the experiment. B. To communicate results quickly. C. To ensure consistent results by repeating the experiment. D. To avoid drawing conclusions. Purpose of evaluation step in scientific method: to ensure constant results by repeating the experiment. Which of the following is the first step of the scientific method? A. Hypothesis. B. Conclusion. C. Observation. D. Experiment. Which of the following is the first step of scientific method? First step, observation. 26. A researcher conducts an experiment to test the effect of a new fertilizer on plant growth. They set up two groups. Group A receives the new fertilizer and Group B receives the standard fertilizer. All other conditions like light, water, temperature are kept constant. After several weeks, they measure the plant height in each group. Which of the following statement best describes the variable the experiment? Which of the following statement best describes the variable in an experiment? A. Plant height is the independent variable and then the type of fertilizer is dependent variable. B. Type of fertilizer is the independent variable and the plant height is the dependent variable. C. Light, water, and the temperature are independent variables. D. There are no control variables in the experiment. Statement best describe variable in the experiment. Type of variables, type of fertilizer is independent variable and the plant height is dependent variable. What happens to a hypothesis after it has been tested many times and found to be accepted? What happened to a hypothesis after it has been tested many times and found to be accepted? It remains a hypothesis, it becomes a theory, it becomes a law, it is discouraged. It becomes a theory. Why is communication important in scientific method? Why is communication important in scientific method? A. To keep scientific discoveries secret. B. To confuse other scientists. C. To share knowledge and avoid repetition of work. D. It is not important. Communication. C. To share knowledge and prevent repetition. Which type of microscope is particularly important for studying fine structure in living cells? Fine structure in living cells. Electron microscope, dissecting microscope, phase contrast microscope, simple microscope. Fine structure of particular. The answer must be phase contrast microscope. 13. What is the importance of reporting and publishing results in scientific research? A. To keep the knowledge secret. To keep the knowledge secret. B. To avoid repetition of the same work. C. To confuse other scientists. D. To make the results look more complicated. To avoid repetition of the same work. Which of the following statements correctly differentiates between a phase contrast microscope and an interface microscope? A phase contrast microscope uses electron beams, while an interface microscope uses light waves. B. An interface microscope uses increased contrast through interface of light waves and can detect smaller differences in contrast and provide color effects, unlike a phase contrast microscope. C. A phase contrast microscope is used for 3D imaging, while an interface microscope is used for living cells. D. There is no difference between phase contrast and interface microscope. They are the same. Which one of the first correct differentiate between the phase contrast microscope and an interference microscope? An interference microscope increases contrast through interference of light waves and can detect smaller differences in contrast and provide color effect, unlike a phase contrast microscope. The last question, but not the least one, is what is the purpose of a transition electron microscope? A. To study the surface of object in 3D. B. To magnify living cells. C. To reveal the innermost detail of the cell interior. D. To view large specimens. What is the purpose of transition electron microscope? Purpose of transition electron microscope: to reveal the innermost detail of the cell interior። ይህን ይመስላል ማለት ነው። በአጠቃላይ 32 ጥያቄዎች። ግልጽ ያልሆናችሁ ነገር ጥያቄ አስተያየት ካላችሁ ኮሜንት ላይ መጻፍ እንዳትረሱ። በቀጣይ በሌላ ፓርት በሌላ ምዕራፎች የምንገናኝ ይሆናል። ቪዲዮን ላይክ ለጓደኞቻችሁ ሼር ማድረግ እንዳትረሱ። ፒዲኤፉን ቴሌግራም ላይ ታገኛላችሁ። ኔክስት እንገናኛለን። ባይ ባይ።